I'm Isabel Fernández. I'm a PhD student for the Translational Venomics in IMDEA Food Institute. And I'm going to talk about my research project that is focused in the antiproliferative profile of an octopus venom-peptide. So first, I want to make a short introduction about the type of cancer in which our peptide has its effect, and this is melanoma. Melanoma is our proliferation of melanocytes, that are the cells that synthesize the melanin, due to a progressive accumulation of mutations that lead to cell cycle deregulation, and that is the deadliest form of skin cancer worldwide, because although in the earliest stages of melanoma is treatable by surgery removal, when it becomes advanced or metastatic, it becomes lethal. So the most common, the majority of the melanoma cases are associated to acquire somatic mutations, and the mutation that is the most common is substitution of valine for a glutamine in the codon 600 of the kinase BRAF. Actually, the standard of care of patients that has this advanced BRAF melanoma is the use of inhibitor of BRAF and MEK, in combination sometimes with immunotherapy. The problem is that these target therapies has been related to the development of secondary resistance that are followed by rapid and deterioration and death. So for this reason, and the fact that the incidence of melanoma is progressively increasing, there is a need of new therapeutic strategies and more efficient targeted approaches. So here is where Venomics appear. Venomics is the research field that is focused in the characterization and study of the properties of animal venoms and their potential use as therapeutic strategies. And as you may know, venoms are mixtures of bioactive compounds that are called toxins. The toxins can be proteins and peptides that can act alone or in combination with others, and can alter multiple biochemical pathways. And for this reason, they have attract the they have attract the like the interest for the pharmaceutical industry, because they see the venoms like a novel source of bioactive compounds that can be used for drug development. Some examples of approved drugs that comes from animal venoms are captopril or exenatide. But it's true that venomics is also a novel field, and there are a lot of venoms from different species that are poorly characterized. And one example of these species are cephalopods. There are many species of cephalopods that produce venomous secretions, both for defense or depredatory mechanisms, and these venomous secretions are produced within specialized posterior secretory glands that the cephalopods have, and the secretions are compound has bioactive compounds that can be both small molecules or peptide toxins. An example of these peptide toxins are the tachykinin peptides. The tachykinin peptides are neuropeptides that are widely found in vertebrates and invertebrates nervous system, and that share a common amidated C-terminal sequence that is important because it's needed to activate the receptors. The receptors of these peptides are called tachykinin or neurokinin receptors, and they are important because once they are activated, they promote the proliferation and migration in tumor cells. So some examples of these tachykinin peptides are OptiK1 and OptiK2 from the Octopus vulgaris, and another example is OptiK3 from the Octopus carna. The important for our research is this one, the OptiK3, because we obtain our peptide that is called OTPET1 from it. OTPET1 is a deaminated form of the OptiK3 peptide that has a highly negatively charged N-terminal and that has a deaminated C-terminal that contains a tachykinin-like domain. So why is interesting OTPET1? OTPET1 has the ability of reduce the BRAF melanoma cells viability. Here you can see that the treatment of OTPET in melanoma cells that is in red is dose dependent, but it doesn't affect in the same way to the healthy control cells that we use that are fibroblasts called NFF. So although the tachykinin peptides are supposed to bind to the receptors and promote the tumor proliferations, our group have seen that OTPET1 has the opposite effect. So we also have studied if this effect of OTPET1 is specific of melanoma cells that have the BRAF mutation, and we have tested OTPET in some cell lines with this kind of mutation. Here you can see that it also has the same effect, reduce the melanoma viability, but when it's used in cells that doesn't have the mutation in BRAF, but another different mutation, it loses the effect. So OTPET1 is specific for this kind of melanoma cells mutations. Moreover, OTPET1 not only reduces the melanoma viability in cells, but it also reduces it in vivo, because we have seen here that nut mice that are treated with OTPET in the tumors lose the volume in comparison with the mice that are treated with vehicle. And not only that, but we have seen also that in several fish xenografts, OTPET1 reduce the viability of melanoma cells in comparison with the treatment of vehicle. So we wanted also to study the function of each part of the peptide, and for that we create two truncated forms of the peptide, and we tested the ability of N-terminal and C-terminal, and we saw that the C-terminal domain has the active site, because here you can see that the C-terminal by itself still have a cytotoxicity against melanoma cells, but the N-terminal controls the potency and the selectivity, because here you can see that the C-terminal without the N-terminal in the sequence also is cytotoxic for the fibroblasts, for the health control cells. So maybe the N-terminal region is controlling the binding affinity of the C-terminal to the possible targets, or maybe it's controlling the internalization of the peptide in the cell. So we also wanted to see if OTPET1 can enter into the cell, or if is just making the effect from outside. So we tagged OTPET1 with fluorescein, and we treated melanoma and fibroblast cells with it, and we saw that OTPET1 enters in both melanoma and fibroblast cells, but the distribution was different. Here in melanoma cells, OTPET1 is localized near to the perinuclear area, but in fibroblast is homogeneously distributed all along the cell. So another important fact is that OTPET1 doesn't cause the apoptosis in the cells. So OTPET1 is fighting the tumor growth by inhibiting the cellular proliferation instead of killing the cells. And another important thing is that the cells that are treated with OTPET1 present higher levels of ROS, of reactive oxygen species, that produce DNA damage, loss of the proliferative capacity, and can induce cellular senescence. So maybe these higher levels of ROS can be the cause behind the biological effect of this peptide. So we next finally realized a proteomic characterization of OTPET1, and we saw that the actin cytoskeleton, the PI3K-mTOR pathway, and EIF2 functions were the main pathways that were downregulated in the cells that were treated with our peptide. And here you can see in this box in green are the proteins that are downregulated in OTPET treatment in the mTOR pathway. So what we did to test some of these things was try the effect of different inhibitors from the PI3K-AKT-mTOR pathway and the MAP kinesis pathways in combination and alone with OTPET1. And we saw that rapamycin, that is an mTOR C1 inhibitor, and LY32149X6, that is an ERK MAP kinase inhibitor, in combination with OTPET1, potentiate the effect in comparison with the OTPET1 treatment alone or these inhibitors alone. And moreover, important is that the combination of these inhibitors with OTPET didn't affect the control health cells. So these results highlight the potential that OTPET1 has as an anti-tumoral therapy in combination with another kind of actual therapies that is safe and can fight against the BRAF mutated melanoma cells. So to sum up all of this data, we have that OTPET1 is effective against BRAF mutated cells in vitro, but it also is in vivo because it reduced the tumor growth in mice and in zebrafish xenografts. We now know that the C-terminal domain is the active part of the peptide because it's the responsible for the cytotoxic effect, and the N-terminal modulates the potency and the selectivity. OTPET1 is a P3K-AKT inhibitors, and when we use it in combination with mTOR or ERK inhibitors, the antiproliferative profile is potentiated. And we know that the mechanism of action of OTPET1 is different from other kinds of venom-peptide that present this kind of cytotoxicity, because not only because of the higher levels of ROS, and that it also alters the metabolic profile of the cells, but it does in another way that is not apoptosis, because it doesn't kill the cells. So from now, the future work will be focused on the characterization of OTPET1 biological activity and the mechanisms of action. We want to identify the target of OTPET that helps to OTPET1 to get this antiproliferative profile against the BRAF mutated melanoma cells. And we also want to examine the efficacy and the safety that OTPET can have alone or in combination with another kind of therapies as a cancer treatment. Here I present my group, the Translational Venomics group, that is made up by Dr. Maria Economopoulou as PI, the postdoc is Dr. Javier Moral-Sanz, and me. And thank you, all of you, for listening.
Thank you very much, Isabel. So are there any questions directly out of the audience? Then you can feel free to ask them directly. Else I would start. Do you have any idea what the biological function might be of the peptide?
The biological function?
So why it's in the venom?
The peptide itself is a tachykinin protein, so it has the effect that the others, and I suppose they bind to the receptor and activate the different function of the tachykinins.
Ah, okay. And did you try some variation of the amino acid sequence? Because it's
yes.
few amino acids, so it might be interesting to vary.
Yeah, we have made different truncated forms, not only the N-terminal and C-terminal, and we have also changed the hydrophobicity of the sequence and see what can be more effective or less. Yep.
Okay. Igor, you have a question?
Thank you, Mike. Isabel, very nice work, very nice presentation. I missed, is this BRAF a receptor, extracellular receptor of these melanoma cells, or?
The BRAF is
yes.
a kinase, is a serine threonine kinase.
So as I understood, this peptide is internalized and it acts intercellularly.
Yeah,
That's
it enters the cells.
Okay.
Enters the cell, yeah.
Do you have some idea how this molecule internalizes?
No, actually, we are working on that. We want to to understand how can enter the cells. Maybe if the N-terminal sequence has something to say. I mean, maybe it's important because, as I have shown, when the N-terminal is present, the peptide is more specific, affecting the melanoma cells than the control healthy cells. So maybe it's something about the sequence of the cell, but I don't know how.
So you think that this N-terminal part, which is negatively charged, is important for internalization?
Yeah. Maybe the charges are important in lipids composition of the melanoma or the control cell membranes.
Yeah, because on the other side, this is a little bit weird that this peptide, which is in fact highly negatively charged, is interacting with a mutant which is negative, which has a mutation valine to aspartic acid. So that would be a little bit strange. Okay, it's not necessary that this is in a direct contact, I mean, in a binding site, but anyway, there is one would expect some repulsion between the similarly or equally charged parts of the molecule. And maybe another bit naive question. Is it difficult to extract the venom from this beast?
Actually, we have get a way to synthesize it chemically, so we don't need more the venom. Yeah.
But if you would need the venom, is it difficult to obtain it from the octopus?
I have no idea.
You mentioned that this peptide is amidated.
Deamidated, yeah.
Do you have amidated, I mean, this synthesized peptide or not? Because on your slide, it was without amidation of the C-terminal carboxy group.
The amidated form is the native peptide.
So you don't work with the amidated form?
I don't usually, no, I don't work with the native form, no. I just work with the output one, yeah.
This is sometimes very important. So probably you tested that the natural amidated form behaves equally than the synthetic one, which is not amidated. Did you check this?
I haven't, maybe in my group before, yes, but maybe no.
Thank you, Isabel. Very nice. Nice presentation.
Thank you.